Hello, and welcome to our presentation on the health benefits and toxicity in tea and kombucha, presented by Brian Martinez, Zachary Rad, and Catherine Wu from the College of Liberal Arts and Sciences Department of Chemistry at the University of Florida. Tea has long been consumed for its alleged positive effects and remains a common drink today. And with increased interest in functional drinks, or drinks that provide some sort of nutritional benefit or value, the kombucha market has been thriving in recent years due to their alleged health benefits. With this research, we aim to provide more information regarding these drinks that have been cited by so many as beneficial for human health. This research has the potential to work in tandem with clinical trials and medical research to determine the beneficial effects of tea and drinks derived from the tea on the human body. We also hope to work with UF faculty in order to create an accessible, comprehensive database of medically relevant information of teas and kombucha. This database can be used to promote public education and awareness of their nutritional information and possible toxicity. The increased interest and popularity in these drinks means it is more important than ever to perform a comprehensive evaluation of the contents, possible health benefits, and toxicity of tea and kombucha. Our focus in this research project is the possible toxicity and medical relevance of tea components. Toxicity can be evaluated via a metal analysis. Heavy metals, when accumulated, can contribute to cardiovascular, nervous, kidney, and bone diseases. These heavy metals can accumulate in multiple steps of the growing process of teas, and even in the fermentation process of kombucha, if fermented improperly. We also aim to examine a wider variety of metals, such as rare earth elements, to fill the gap in the literature. On the other hand, potential health benefits have many more different aspects and will be examined using many different methods. Polyphenolic compounds in tea, such as catechins, theaflavins, tannins, and flavonoids, have been related to various health benefits. The probiotic effects of the microbes present in kombucha must also be further examined. The fermentation process that yields kombucha will also be analyzed and its effect on the polyphenolic compounds present in the tea it was brewed from. The microbial composition of kombucha is also claimed to add to its potential health benefits, and this must be further examined. To construct a comprehensive evaluation of the potential health benefits and toxicity of tea and kombucha, we utilized 6 different instruments to fill the gap in current literature: Fourier transform infrared spectroscopy, ultraviolet visible spectroscopy, fluorescence spectroscopy, atomic emission spectroscopy with inductively coupled plasma, nuclear magnetic resonance spectroscopy, and liquid chromatography with mass spectrometry will be used to further examine the contents of teas and kombucha beyond what has been researched so far. Multiple brands of tea and kombucha will be used to provide a thorough review for customers, as well as evaluate the differences between brands of the same type of tea. Both home-brewed and commercial brands of kombucha will be evaluated for consumers who are interested in brewing their own kombucha at home, as well as consumers who are more interested in the nutritional value and benefits of kombuchas already in the market.
To begin our research project, we first need to understand the previous characterization of polyphenols in tea. Both green and black tea contain about 30 to 40% polyphenols by weight, and we're particularly interested in these polyphenols because many of them have been shown to contain bioactive properties. The main difference in the preparation of green and black tea is that during the manufacturing process, they undergo different degrees of oxidation, which overall leads to them having different chemical composition as well. So, for example, in green teas, because they only undergo minimal oxidation processes, we are able to see a greater concentration of monomeric polyphenols like catechins. Black teas, on the other hand, undergo a more rigorous oxidation process, and this causes their polyphenols to often become oxidized or polymerized. And we can see this in the case of compounds like thearubigens and theaflavins, which have a much higher concentration in black tea than in green tea, although black tea does still have a certain concentration of catechins as well. These polymeric polyphenols are a little bit more difficult to understand because their bioactive properties have not yet been determined. In the table to the right, we can see an approximate breakdown of what polyphenol compounds are present in green and black tea, as well as some other organic compounds and inorganic materials that are found in these teas. Now let's take a look at some of the potential health benefits of these polyphenolic compounds found in tea. Compounds in green and black tea have potential applications in treating a variety of societally relevant ailments. These include cancer, obesity, oral disease, and many more. To give just a couple of examples, theaflavin-3 gallate and epigallocatechin gallate, which are primarily found in black teas and green teas respectively, have been shown to prevent the formation of carcinogenic heterocyclic amines, and that is just one of the potential anti-cancer applications of polyphenols found in tea. A second example comes from the treatment of cardiovascular disease. Black tea extract and epigallocatechin gallate are able to oxidize low-density lipoproteins, and this is very important to treating cardiovascular disease because these LDLs are able to build up on the interior of blood vessels. By oxidizing them, we are able to prevent this buildup, and that shows just one more of the potential health applications of our research. Our characterization of the flavonoids in tea is also informed by previous research studies. For example, Li et al. Was able to use ultra-performance liquid chromatography diode array detector coupled with quadrupole time-of-flight mass spectrometry to separate and identify 24 flavonoid derivatives from brewed teas at various stages of the manufacturing process. Li was also able to compile an MS library of 64 flavonoid derivatives, which will be useful in identifying the compounds we detect in our tea samples. Konieczynski et al. Used high-performance liquid chromatography with an ultraviolet detector to quantify the concentration of 5 flavonoid derivatives in tea samples. For example, on the chromatograms we see on the right, we can observe for both A, green tea, and B, black tea, 5 resolved peaks corresponding to distinct groups of flavonoid derivatives. Li's and Konieczynski's experiments provide the basis for our methods in LCMS and LCUV, which we will elaborate on later in the presentation. We have also looked to previous methods used in elemental analysis, and in particular metals analysis, of tea samples. Ma et al. Used inductively coupled plasma mass spectrometry to determine the concentration of many metals, including both essential elements and toxic heavy metals. In addition to the use of ICP-MS, Ma employed multivariate analysis methods like linear discriminant analysis to differentiate tea samples grown in different regions, which can be shown in the scatterplot of discriminant scores. Besides ICP-MS methods, Islam and Ebihara have been able to characterize Japanese green tea leaves and tea infusion residues using neutron-induced prompt gamma ray analysis, or PGA, and instrumental neuron activation analysis, or INAA, and the elemental characterization of Japanese green tea leaves. The primary practical difference between the Ma and Islam methods is that PGA and INAA are non-destructive and require little sample preparation. However, because of the ease with which we are able to obtain and prepare samples, these factors are not high on our priority list, deeming the ICP method sufficient. Similar methods have been used in looking at the chemical and elemental analysis of kombucha. One difference in the polyphenol composition of kombucha versus green and black tea is that we notice an increase in the overall polyphenol concentration across the fermentation process. However, during microbial fermentation, polyphenols demonstrate variable chemical stability, with many undergoing chemical changes. This provides an interesting research pathway into polyphenol chemistry across microbial metabolism in the acidic kombucha environment. Bauer-Petrovska and Petruszewska-Tosi have further been able to use atomic absorption chromatography and elemental analysis of kombucha, spanning both essential elements and heavy metals. Kombucha also has an important point of difference from green and black teas in its microbial composition, which may also have health implications due to the purported probiotic properties of kombucha. Al-Adami was able to use fluorescence spectrophotometry in analyzing the viable cell density in kombucha and other microbe-containing food products. Now we'll take a look at the analytical methods we will be using in our project, starting with fluorometry. The primary purpose we will be using fluorometry for is to determine the density of viable cells in kombucha. Al-Adami used a microfluidic chip as the sample holder in which to measure the fluorescence of culture to which resagirin is added. However, we will be using an adjuvant photometer and quart sample holders for the same purpose. How this method works is that enzymes in viable bacterial and eukaryotic cells are able to reduce resagirin to resarafin, which is fluorescence active. The greater the cell density, the more quickly we will observe the increase in fluorescence, which Al-Adami showed can be modeled using a linear relationship. Through the slope of this line, we can obtain a measure of the viable cell density. To prepare our samples for fluorometry analysis, we will inoculate 3 milliliters of kombucha into 47 milliliters of LB media. We will then incubate this culture at 37 degrees Celsius for a period of 2 hours. Following that, we will add 5 microliters of resagirin to a 1 milliliter aliquot of the culture sample. Immediately following that, we will do fluorescence analysis to obtain the viable cell density. For the elemental analysis, we will use inductively coupled plasma atomic emission spectroscopy, or ICP-AES. We will first digest the tea infusion in 5 milliliters of nitric acid and add lanthanum to be used as an internal standard. Once digestion is complete, we will dilute the samples to 12.5 milliliters using 2% HCl. By coupling the ICP to the AES, we hope to identify characteristic ion peaks across different wavelengths in our atomic emission spectrum. This will give us both the elemental composition from the peak position and the concentration from the integral. An example of ICP-AES peaks being used for this purpose can be seen in the 3 distinct peaks to the right. We will use Fourier transform infrared spectroscopy after separation by liquid chromatography to confirm the presence of phenyl groups in the flavonoid derivatives to ensure the correct selection of peaks to be quantified using nuclear magnetic resonance. This will also allow us to compile an FTIR library of flavonoid derivatives, which can be made available to other researchers to provide new pathways in terms of flavonoid research. An example of the type of FTIR results we are aiming to obtain can be found in the spectrum to the right, which shows an FTIR spectrum for EGCG nanoparticles.
Next, we will also be using nuclear magnetic resonance spectroscopy for our analysis. NMR spectroscopy is used in research for quality control and to assess purity. It allows chemists to be able to determine molecular structures and functional groups as well. A lot of research of tea analysis has been conducted in the past using NMR spectroscopy, but we saw this as an opportunity to expand NMR research on kombucha analysis to observe more flavonoid derivatives from black and green teas that have yet to be studied in previous research. With NMR spectroscopy, we want to verify the structures of the flavonoid derivatives present in our samples. And it will be used to confirm HPLC analysis. Here is an image of the Bruker 600 MHZ NMR instrument that we have chosen to help us carry out our analysis. And for our results, here on the left is an example of an NMR spectra of the flavonoid compound daidzein. Different functional groups are present throughout this spectra. And here on the right are structural formulas of various flavonoid derivatives that have been found in previous tea research. Here is another example of an NMR spectra with various functional group peaks along with their concentrations. We will use this to determine the structure of the different catechins in our sample and make comparisons between the different types of tea used in the samples. Liquid chromatography and mass spectrometry will be used in the fragmentation of different compounds to discover which of the 64 flavonoid derivatives from our standards are present in our tea samples. We will be using an organic solvent like diethyl ether for our analysis with an Acentis C18 15 cm column in negative ion mode. This instrumentation will be paired with UV-vis spectroscopy to confirm flavonoid presence. I couldn't find a picture of the Bruker UHPLC UVMS that we chose for our analysis, so here is a picture of a UHPLC MS thermo instrument instead. And below it is the Acentis C18 15 cm column. We will be using the retention time graphs to compare the retention times of our known sample, our standards, and our unknown sample, our T samples. If there is a peak for our sample that is in the same location as the standard, then a positive identification can be made based on retention times. And here to our left is an example of retention times in minutes of flavonoid compounds. And here is an example of a spectrum with various flavonoid fragments separated by their MZ values. Intensity is measured in percentages up to 100 as seen here. Now for ultraviolet visible spectroscopy. The HPLC-MS will be coupled with UV-vis spectroscopy to determine which peaks absorb in the range expected for polyphenols and are likely to contain flavonoid derivatives. We will be using this to test one of our hypotheses as well. The hypothesis states that the acidic environment produced by the kombucha microbiota will decrease the amount of catechins. We will observe catechins and quantify them by using UV-vis spectroscopy and liquid chromatography mass spectrometry. And now for our conclusions. Our overall goal for our project is to make a comprehensive comparison of the potential health benefits and toxicity of green tea, black tea, and kombucha tea derived from these teas using qualitative and quantitative data from our various methods. We want to provide society and the scientific community with our findings so that they can make informed decisions when they are looking into which type of tea to choose, and to open the door for more research on this to be done. We also aim to confirm or reject our hypotheses. We hope to determine if 1. Metal toxins isolated from the kombucha reflects which type of tea it was fermented from. We also hope to determine if 2. The acidic environment produced by the kombucha microbiota will decrease the amount of catechins. And lastly, 3. If the fermentation conditions of kombucha increase the amount of metal toxins present using our various instrumentation methods. And for our acknowledgements, we would like to thank the National Science Foundation and Dr. Alexandra Jacobs and our lab teaching assistants for all of their guidance throughout this course. Thank you.